Is it working? Yep.
All right. Continuing on with signaling pathways. We're going to look briefly at two more signaling pathways as both representative examples and ones that are actually quite important. Here's another signaling pathway that involves a certain very huge diverse family of growth factors, but it uses a very different pathway than the RAS-MAPK pathway. This pathway, in fact, does not use any G proteins at all. There's no G proteins at all in this pathway, but it's still a very important one. And this one is called the transforming growth factor beta pathway, or TGF-beta is a short thing for it. Now, what is a transforming growth factor beta? Okay, well, it's actually a very huge, diverse family of growth factors that are very important in all kinds of different developmental events, all the way from early embryonic life all the way up to the late stages of organ formation. Now, the term transforming in cell biology, when you say you transform a cell, what you're doing is turning a cell into a cancerous or cancer-like cell. Now, obviously, we're not going to have stuff like this that is out there to turn cells into cancer cells. It's not a good idea, trust me. But that was the first thing they noticed when they took this stuff and applied it to certain cells. But in real life, it does a whole bunch of things, causing cells to proliferate, causing cells to differentiate, but it uses a very different pathway than the RAS-MAPK pathway. It's a huge, diverse family of growth factors in a number of categories. The TGF-beta, there's quite a few of these guys. There's another family called the activins. The activins are very important in determining in an early age what's going to be the ventral or belly and what's going to be the dorsal axis. They play a very big role in that. Another big subfamily within this is called the bone morphogenic proteins, or BMPs. Now, obviously, with a name like that, they play a very critical role in organizing your skeleton. But that's not the only thing they do. They were first discovered because they play a critical role in skeletal formation, but they do all kinds of other things. They're involved in limb formation in general, not just the skeletal part of it, but the rest of it as well, and many other kinds of things. So these are three subfamilies in this very large, diverse family of growth factors. And once again, many cells secrete these things, many cells respond to one or more of these different members. And here again, we see these involved in all kinds of developmental events. Now, the thing about them is they all use a common signaling pathway that we're going to take a look at in just a moment. I guess a few people are still writing people. Excuse me? MOR, morphogenic, that means structure formation. Okay? So that's why your skeleton looks the way it does. You think these guys. Yeah, these are subfamilies within this big, huge superfamily. Take a look at this pathway here. Once again, the neat thing about this pathway, unlike the others we've looked at, does not use any G proteins at all. Instead, we're going to directly activate a family of transcription factors. Let's see how this thing works. It turns out there's receptors for the TGF-betas fall into two categories. They're quite similar to each other, but we have something we call a type 1 and a type 2 receptor. They're slightly different from each other, but they do have a lot of similarities. So what happens first, we're going to have a type 2 receptor here, and it's going to bind the appropriate TGF-beta. Once that happens, that will allow the related type 1 receptor, which will bind to both. So now we've got two receptors bound to our growth factor, the type 2 receptor and the type 1 receptor. That binding to each other causes structural changes in these proteins. And it turns out, once again, these are protein kinases. They phosphorylate target proteins. But they're not the tyrosine kinases. They phosphorylate proteins on the usual amino acids, serine and threonine. So they're regular type protein kinases. What happens when we get this together is we turn the kinase activity of the type 2 receptor on, and what it does is it phosphorylates the type 1 receptor. So the type 2 receptor's target is the type 1 receptor. Once you phosphorylate the type, and I'll put a star here saying that this is active kinase, once we phosphorylate the type 1 receptor, its kinase activity is switched on too. And it phosphorylates target proteins. Once again, we're seeing the vast importance of protein kinases and phosphorylation to regulate protein activity. Now, when this guy's active, what it does is phosphorylates a family of gene-regulating proteins or transcription factors. Remember the term transcription factor from genetics? It's a protein that binds a section of DNA in front of a gene and turns it on or turns it off, regulates transcription. Well, this guy phosphorylates and activates a series of transcription factors, a family that's called SMADs. I don't know what that stands for. It's an acronym for something. But it activates these guys, phosphorylates them. So we'll put transcription factors here. And there's a family of these guys. So each cell has one or more of these guys. Now, once it's phosphorylated, it does two things. One of it undergoes a structural change and exposes what we call a nuclear import sequence. It's a stretch of amino acids that act as a ticket to the nucleus. So it's just like if you're trying to go to a rock concert or something, they're not going to let you, they're not going to let you in unless you got the appropriate ticket. So now we have this ticket to the nucleus. So this protein will go into the nucleus. Once it's in there, it binds to a relative that's always in the nucleus that's called SMAD4 or co-SMAD. And this assembly then latches on to DNA and regulates a whole battery of genes. Many of these genes are important in regulating, in causing cells to differentiate, causing cells to proliferate, and things like that. Yeah, or co-SMAD is another term. You'll see both. Okay, now, so that complex regulates whole bunches of different genes in different cell types. And each one of these SMADs is going to deal with a different set of genes. So that's why you can get different responses with the same growth factor in different cell types. It depends on what you got down here, what ones you have here. So in this sense, the amplification step is going to be phosphorylation of a large number of these transcription factors. And clearly these things regulate large numbers of genes, so they're used in all kinds of different developmental events. Those of you who take 413 with me this spring. Anybody going to take 413 with me this spring? Okay, we're running these guys again. Development, animal development. Okay. All right, so that's yet another signaling path. We're going to take a look at one final one that's also very important development, and its regulation is quite a bit different. Because in effect, in this particular signaling pathway, what happens is a very important transcription factor is normally sentenced to death by proteasome. That's a horrible thing. They got to ban the Supreme Court out ban that. That's a horrible way to go. You know, death by proteasome. Well, ordinarily, it's under a death sentence. But activation of a particular receptor is a state of execution for that protein. So what we're going to see here is an example of protein regulation by destruction in the proteasome or by preventing that destruction. And that is a signaling pathway known as the Wnt. Now the granddaddy of this is a gene called wingless that they first discovered in fruit flies. And it's called that because fruit flies with that mutation were normal except for one thing. They never grew wings. It makes it real bad for a fruit fly. But later on they found that vertebrates have numerous different members of this. This is a large family, a dozen or more different proteins. Now, what's going to happen here is we've got a receptor.
Actually, 3 receptors though.
Got a little senior moment here. I forgot the name of that receptor. There's 3 different receptors. One's the major one. But let's just say receptor for time being out. The receptor for these particular proteins. Okay, now, what's going to happen ordinarily is there's this very important transcription factor called beta-catenin. And in the absence of our the Wnt are small signaling molecules, much like growth factors, small proteins, and they're secreted by a number of cells. Let's say in this case we have no Wnt proteins around. Okay, we have this transcription factor called beta-catenin. It's assembled with a couple of other proteins that we don't need to know about. But ordinarily, in the absence, here we have an empty receptor, no Wnt's around right now. What's ordinarily going to happen is you have a kinase that binds to this and phosphorylates the beta-catenin. When that beta-catenin is phosphorylated, it's recognized by one of these ubiquitin ligase proteins. It recognizes the phosphorylated version, and you know what these guys do. What these guys do is they covalently attach ubiquitins to it. And then that means you go into the proteasome and die. Okay, so in the absence of the Wnt signaling molecules, beta-catenin gets phosphorylated, tagged with ubiquitin, and dies a horrible, nasty, painful death in a proteasome. So, of course, a transcription factor that's destroyed is not going to be able to regulate too many genes. Now, what happens, though, in the presence of the Wnt's, our signaling molecules, that gives a state of execution. Because through a particular pathway, what's going to happen is this kinase here gets inhibited. The receptor activates a particular protein, and that protein goes and inhibits the kinase, so beta-catenin never gets phosphorylated. So beta-catenin, if it's not phosphorylated, the ubiquitin ligase doesn't even know it's there. So it never gets ubiquitins attached to it, and that means it gets a state of execution. Now, in this case, what happens, it goes into the nucleus. And once it's in the nucleus, it's going to regulate a whole bunch of different genes. Now, beta-catenin plays critical roles in numerous developmental events. In echinoderms and vertebrates, it's one of the major factors, along with some of the TGF-beta family members, but it's one of the most significant factors in determining all the way in an early embryo what is the dorsal or back and what is the ventral axis. One of the first things you have to do in an early embryo, you start out as a nice spherical egg, is you have to say, this is belly, this is back, this is head, this is tail. You've got to set those axes, the anterior, posterior, and the dorsal ventral axis. The WNTs through beta-catenin do that. Beta-catenin usually activates things that are involved in ventral tissues and stuff like that. Very early in embryonic life, the WNT signaling pathway and beta-catenin is going to determine what is the ventral or belly part of the early embryo. As development proceeds, you get many different kinds of tissues forming, and here again, most of them have a dorsal ventral axis. Think the spinal cord. This is a good example. The dorsal part of the spinal cord is significantly different than the ventral part of the spinal cord. You can thank the WNTs for providing that. If you have mutations in there, you get all kinds of abnormal spinal and brain development. You literally don't know what's up and down when that happens. And numerous other tissues, beta-catenin determines, through the WNT signaling pathway, determines what's the ventral or belly part of that tissue or organ. So it plays numerous roles in development. And this is an example here of protein regulation by destruction in proteasomes. And in this case, we're not actually seeing G proteins or anything like that. It's either allowing the death of beta-catenin or sparing its life so it can go and regulate genes. Okay, questions on those two pathways? I wish I knew. I wish I remembered what the main receptor for that was. I know the other two, that's the negative part. But the main one I just forgot. Sorry about that. Okay.